Welcome to another video of Stat 212. So today we're going to introduce the idea of design and talk a little bit about the components of an experiment. So design is a really important element of doing statistics, of trying to create and understand data, because design is kind of where it all starts. Like we have data, we see data, we work with data, but that data came into being from some source, and that's going to relate to the design. How is that data collected, and what kind of experimental setting or non-experimental setting produced that data. So fundamental to this conversation is this idea of validity that we're going to talk about, and we're going to talk about it throughout this entire unit. But validity is fundamentally this idea of how strong is this study, and what kind of claims can I make from it. So validity is concerned with questions like who was in the study, were outside influences controlled, or did I just kind of observe data in the wild and I have no idea what's causing what? What was measured and how was it measured, right? So what choices were made in terms of instrumentation? Where and when was it conducted? Those are all questions that we need to answer in order to understand how strong this design is and what kind of claims are really appropriate to make from this data. And so validity is usually kind of a hard thing to know at a surface level, because usually when we're encountering data, and especially like we're reading articles and we're reading reports, what we get are the claims, and we might get some information about, say, the sample size, or maybe a little bit about who was in the study, which, who was in the study is helpful. The sample size, not so much. Sample size doesn't really tell us about validity, which we'll talk about later, what it does tell us, and it's not exactly validity. We might get information about like p-values and sample statistics, but again, those don't tell us anything about the validity of the study. So usually we have to dig a little bit to understand. And so kind of that first step of digging, that first layer, is what was the design of this study? Was it an experiment or an observational study? And even between those two, what type of experiment was it? What type of observational study was it? And so that's what we're going to kind of focus on in these first few pages. So I mentioned experiments and observational studies as kind of like the two types of studies that we're looking at. And these are both studies, we would call them analytic studies, meaning that the goal is to look at a relationship between two variables. And most of the time, we're interested in whether or not there might be some kind of causal link. But as we'll talk about with validity, sometimes the design is set up well so that we can be pretty confident in a causal link. Sometimes the design is not so good, and we actually don't know what's causing what, or if there's something else that that we didn't directly measure that's interfering, and that's involved here. But kind of that first stage is, what kind of study was done. Experiments are usually better at determining causal links. Observational studies, it's a little bit harder. They're usually not very good at establishing causal links. Two other types of designs that we don't really talk about in this class would be a descriptive study, and that would usually be, usually those are more univariate studies where we're just kind of looking at, we have some data and we're just reporting information about it, right? It's like we're just reporting descriptive statistics, and that is the end game. We're not trying to make inferences. We're probably not trying to necessarily look at relationships between variables. We're just reporting information without making inferences. Qualitative studies would be another type of non-analytic design where I'm not making generalizable claims because I'm not randomly sampling from the population or trying to get a representative sample. I'm just trying to get quality-rich data from usually a limited source. Now, all of these types of studies are still important to do. They all give us data. They give us information, and information is helpful for a lot of different reasons, but we want to be careful that we know what kind of information we're getting and what to do with that. So when it comes to looking at potential causal links, these are the kinds of designs that we're interested in, where this is going to be the best source for causal links. This is going to be more causal suggestions, like ideas and associations that may or may not be causal links, but hey, we can at least look at the relationship between these two variables. These other two types we won't worry about. They give us information in different ways, but not necessarily in establishing causation with the same level of certainty and generalizability as these other two. All right, so we're going to focus on experiments, right? So experiments they are designed for identifying causal relationships. Let's talk about how they do that and what makes something an experiment. So the idea is I have intervenable power. So if it's an experiment, it means that I can, I mean, not like me personally, but, researchers, the team, whoever's involved, can administer interventions. And typically, those interventions are assignable, meaning that I can decide who is getting which intervention. Oops. So that's fundamentally what makes something an experiment, is that we actually have control to some degree about this intervention, including who gets it, and to some degree the environment that intervention takes place in. And the reason why that's so important is because we want to be confident that this intervention is the only difference, like it is the sole cause. Therefore, if I have two different groups and I can give them the exact same experience except this intervention of interest, like this group is given drug A and this group is given drug B or a placebo drug or something like that, but everything else is held constant, everything else is the same, then if I see a difference in the result, if I see a difference in the response, I'm going to feel pretty confident that it's the drug, it's this intervention, and not something else, because everything else was held constant. So there's a lot of things we have to keep into account and understand what makes something a good experiment, but fundamentally, this is what makes something an experiment. So treatment is a term that we use to describe kind of this causal, this intervention. It's the thing that I'm interested in might be causing something. So treatment doesn't always have to be a good thing, though, so don't let the term confuse you there. Like, it could be like a drug. It could be some kind of, literal treatment procedure. It could also be something bad, like, something that might have a bad effect. So smoking could be my treatment factor. Like, I want to understand, causes, or possible responses from smoking as a causal agent. So smoking would be the treatment factor in that study. The response would be the outcome of interest. It would be the effect. So that might be, number of days with a sickness, or, some kind of, outcome or measurement or anything like that, that I'm interested in understanding more about. So then, in experiments, we're typically, not always, but typically we're going to have multiple groups. So often we call them like the treatment group and the control group. If we just have two groups, it's possible we might just have two separate treatment groups, like a treatment A and a treatment B. That's certainly possible. We might have more than two. We might have 3 or 4 or 5 different experimental groups. But fundamentally, just be familiar with these names. That a treatment group implies that it is the group that's getting kind of this treatment of interest, and a control group would be getting either nothing or a placebo or a standard treatment that we already understand how it works, things like that. And the reason we do that is for sake of comparison, right? So if I only had a treatment group, like I had a group of people, and I said, hey, I have this great drug that I think will make you feel better or lose weight or do this or that, and then these people take it, and then I measure the results. Well, am I really sure it was the drug, or was it the experience of being in the study? Was it the psychological effect? Was it something else that motivated them to make other changes? Given that they were taking the drug, maybe they were eating differently too, because they were being cognizant of, like they had a goal in mind. So that's why it's important to have control groups for the most part, is because we need that comparison where we can, kind of even out all these other factors except for the intervention itself. All right, so placebos. Many of you have probably heard what these are, but it would just be some kind of fake treatment that mimics the intervention, and it mimics the psychological effect of having a treatment. So a placebo often would be kind of like a non-effective tablet, like a pill, or some other kind of distraction that will simulate that experience for the control group without having the actual, inherent benefit that we're trying to study. Blinding is another important term that kind of goes along with placebos. So blinding means that the participants themselves don't know which group they're in. Like the participants don't know that they are in the control group or the treatment group, because if they did, then there might be a psychological effect that's confounding this result, right? So it wouldn't just be the intervention; it could just be that, well, these people knew they're getting better, and these people knew that they were just in this stupid control group. So there's a psychological factor that might be influencing the result in addition to the treatment itself. So blinding is important when we have multiple groups like that. Double blinding is kind of another layer to that, and that means that the administrators, the people interacting with the participants, also don't know who's in the treatment or control group. So if you think about some kind of treatment, care intervention, if I have an administrator who knows these people are getting the real thing, they're getting the experimental drug, and these people are just getting a placebo, is it possible that the administrator might act differently around the participants? So that's like another consideration there, that, in some cases, that might be pretty important. In some cases, maybe it doesn't matter at all. In some cases, maybe there is no administrator directly interacting with the participants. But in cases where there are, and in cases where it might matter, it's good to do that. It's kind of another layer of protection on the experiment. So with these ideas in mind, let's go ahead and walk through a practice problem, identify some of these ideas. I'm going to see if I can move my camera here, because I don't need to be in this shot here. And then I probably need to, let's see, is this the right one? Make this show up there. Okay, cool. All right, so mosquitoes find their victims in part by odor. A study looked at whether someone drinks beer or not, might influence the likelihood of mosquito contact. So here in this study, we have 25 participants who drank 16 ounces of beer, and then for each participant, a container holding 50 mosquitoes was released next to that human, and the researchers measured what proportion of the 50 flew closer to the human. They repeated this process with 25 participants who simply drank 16 ounces of water. Okay, so let's identify the experimental units. In other words, who or what is being studied. So the experimental units would, in this case, be the people in the study. So we didn't really define that, but it's important to kind of, just be aware of this, of who are the participants, who are the units that are being studied and measured. And that's going to be people drinking beer or water. In some cases, we might do studies where the experimental units are animals, maybe they're plants, maybe they're something else, right? So we just want to keep that in mind, who or what we're measuring. The treatment factor here is going to be drinking beer. So that is kind of this intervention that has been assigned that we're curious about. And in particular, the motivation for this is the effect of alcohol. Does drinking alcohol, or actually, sorry, a study, I guess it doesn't say, it says odor, but I guess it doesn't specify the odor of alcohol. In this case, they just said, like, we're just studying the effect of the odor of beer. So in this case, the treatment factor is just drinking beer. And we also have a control factor here, and that is drinking water. Now, obviously, we could have just had the control group not do anything. We could have had them drink something else. We could have had them drink non-alcoholic beer. But it really just depends on what is that central comparison that the researchers are trying to make. Is it the odor of alcohol in particular? Is it the odor of beer versus kind of a non-aromatic beverage? Is it just a beverage, period? Right, so there's always going to be instrumentation choices that affect, the strength of the study and what we can and cannot say about it. But the researchers just distinguished beer from a non-aromatic beverage. The response variable here is going to be let's see, it looks like it is the proportion of mosquitoes that flew closer to the human. So proportion. This, by the way, sounds like an awful study to participate in. I hope these participants got paid good money. I definitely would not do this for free, but hopefully these participants had a good incentive there. One thing we don't talk about necessarily in this class is institutional review board um, um, and research ethics, and basically like how studies get cleared and what is ethical and not ethical. So we don't really talk about that, but that's a whole nother conversation. Research is how do you recruit participants? How do you incentivize participants? What's ethical and non-ethical, and how is that decided? And this idea of greater good versus risk and things like that. But anyway, that's completely aside. But yeah, so that's hopefully helpful in just kind of introducing these terms and how we identify them in an actual experimental setting.